Hi, everyone. This is a pre-recording of the lecture for Thursday, October 15th. So we wrapped up on Tuesday talking about unculturable bacteria. I want to emphasize to you that that's a really important point because there are so many bacteria in natural environments that we have not yet been able to cultivate in the laboratory. Um, there are It's not just bacteria. There are lots of fungi, archaea, and tons of viruses that have not yet been analyzed in the laboratory. And so the way that we're getting at that now primarily is through metagenomics. So analyzing the DNA sequences of these organisms or viruses. And in the case of viruses, realizing not all of them have DNA genomes, so we have to look for RNA as well. Okay, so metagenomics is being used to characterize populations to see what kind of genes they have, what kind of functions those genes may encode, etc. So we're not going to talk a lot more about that here, but just to recognize that that's a really important point when we can't culture microbes in the lab, at least with our current technologies. Okay. Continuous culture is an important thing to point out. We were looking at a growth curve in a closed system, right? So we have lag phase, log phase. Eventually you exhaust some key nutrients or you build up waste products, and then things stop, you hit stationary phase, and then eventually things will die. Okay. A closed system does not characterize many natural habitats, right? In most natural habitats, we have materials coming in, materials coming out. They may be on different time scales, they may be slow, but essentially we rarely have a truly closed system. So it's important to think about how microbes are growing in real world habitats in these sort of continuous systems. We may try to mimic that by setting up a continuous culture in the lab where we have a constant input of nutrients and a removal of waste products. If we do that, we can potentially keep microbes growing for long periods of time rather than having just that transient log phase until the nutrients are exhausted. And that's useful to us because then we have more opportunity to study what happens during continuous growth. We also, using a continuous culture system called a chemostat that I'll show you on the next slide, we can actually control the growth rate as well. So we can see what's happening when microbes are growing very slowly versus very quickly, and how different processes going on in the cell adapt to that and are coordinated, right? So a continuous culture in its simplest form looks sort of like this, right? This is our culture vessel here. This is the key thing. This may be stirring. It's hard to do in a shaker, so usually you have a stir plate underneath this and a magnetic stir here, or you have it built into your fermenter. But this is fresh medium, and we have fresh media flowing in, dripping into this. And then, of course, because the volume can't just keep increasing infinitely, we have it dripping out here at a certain level into our waste vessel, right? As shown here, we also have air being supplied in here. All this, of course, has to be sterile so that we don't have contaminants getting in there. So without going into this in great detail, essentially what will happen as you input nutrients in here is that the microbes in this in the growth vessel will grow to their maximum capacity, and then the only growth they can carry out after that is based on whatever new nutrients are coming in. So the flow rate of new nutrients in here is actually determining the generation time of these bacteria or whatever microbes you have in here, right? Because they grow up to their maximum amount, essentially sort of a pseudo-stationary phase, or a steady state is a better way of putting it. And then that steady state, the growth rate in that steady state is determined by the influx of new nutrients. So you can force them to grow slowly, you can make them grow fast. Ultimately, they reach their maximum growth rate, and if you try and get them to grow faster than that, then they start washing out, okay, because they can't keep up with the flux of new nutrients. Okay, so a continuous culture is a very useful research tool. It can be a useful production tool as well for biotechnology. If what you're having, if the microbes growing in here are producing something important, then you can collect them and just basically keep cranking them out. Okay, so the relevance of this system to the real world is if you think about something like the human gut or the digestive tract of any animal, right? So the microbes that are living in the intestines ultimately are limited in growth by the flux of new nutrients coming in. And of course, the waste product is being expelled in feces, right? So it turns out that the residence time in the gut, in the large intestine, say, to be more specific, is about 24 hours, right? Or actually, I should say, it takes about 24 hours for materials to pass all the way through the digestive tract. The bulk of that is in the digestive tract, in the large intestine, right? So at any rate, you know, that culture, an E. coli living in your colon can't have a doubling time of about more than once a day, right? Because if they were growing every 20 minutes, then they would very rapidly, you know, proliferate to fill the whole colon and et cetera, et cetera. We actually are going to do an exercise on looking at what happens if you let E. coli keep growing at its maximal growth rate for various periods of time. We'll talk about that in class. Okay, so that's a continuous culture. Now, the main part of what I wanted to talk to you about is in the, for this part at least, is the last section of chapter 6, section 6.5, which has to do with controlling microbial growth. Because there are many real world situations where we actually want to either kill all the microbes or at least keep them from growing for various reasons. Including, for example, food spoilage, or of course, you know, microbes that are causing infections that we want to kill. Okay, we are going to talk about antibiotics later in the class in the medical micro section, so we're not going to get to that right now. Okay, so one way that we can remove microorganisms is by filtration. And people have actually been using filtration for a long time before they actually knew about the details of bacteria. Water has been purified by filtering through sand and charcoal and other things for many centuries. And that does kind of work because the bacteria will stick to these sort of porous surfaces, right? But it's generally not a satisfactory method for what we're trying to do right now. I should point out that this filtration can also be used for removing larger particulates that are contaminating water. So sediments, you know, dirt, soil particles, etc. So that's another reason why filtration's been around for a long time. But the contemporary methods that we know will get rid of bacteria often use nylon or Teflon filters that have very small pore sizes that are calibrated to ensure that you can get rid of at least all the bacteria if you have a pore size of 0.2 or 0.45 microns, right? So that keeps almost all bacterial cells out, keeps all eukaryotic cells out. It doesn't keep all the viruses out, though. As we know, viruses are generally even smaller than bacterial cells. So if we want to potentially, if viruses are an issue, then we potentially have to use even smaller pore sizes, and that's we call ultrafiltration. Okay. Now, filtration, physical filtration is not a perfect solution because filters can get clogged up, especially if you have a lot of turbidity in the water that you're putting in there. Ultrafiltration especially requires a lot of pressure to push the water through these very tiny pore sizes, and any sort of liquid that has much viscosity is not going to do well through these tiny pores, even if you're just talking about 0.2 or 0.45 microns, right? So pure water is fine, but if you have other components in the water, then, and you have much viscosity, if you have various polymers in there, or if you're trying to filter non-water things, then it's not going to work. Think of like trying to filter something like, maple syrup through a tiny filter. You have to have enormous pressure behind it to get the material to go through. So filtration is useful in some cases, but it's not the only way to do it. Another way to do it is using temperature, right? When we were talking about thermophiles, we talked about the challenges that microbes have in living at high temperature because heat denatures proteins and nucleic acids, right? Things that depend on hydrogen bonds that are not super strong often fall apart when you raise the temperature very much. Boiling water at 100 degrees is enough to kill most microbes, right? So that's like, you often hear about, for earthquake preparedness or something, if there's potential sewage contamination, they say boil your water before using it. Well, that does get rid of it does kill most microbes, but it doesn't kill everything, right? And that's why we have autoclaves. So there are some microbes like hypothermophiles that can survive really high heat. We don't usually worry about them for human health because they're not pathogens, but there are some microbes that form spores. We haven't talked about it much yet in class, but we will later, that are highly resistant to high temperatures. These are generally Bacillus and Clostridia, and there are certainly pathogens in those two genera, right? And they will resist boiling temperatures. So an autoclave raises the temperature even higher to 121 degrees under pressure so that the water doesn't boil away. And that's sufficient to kill, to inactivate even spores. But autoclaving is not a solution for everything. If we're talking about food safety, for example, if you try to, there are a lot of food products that if you stuck them in an autoclave, what would come out the other end would not be something that would be very appetizing, right? It tends to destroy flavor profiles. It tends to affect the integrity of the materials. And so when we do autoclaving in the laboratory, like to prepare media or for tools in the laboratory, they all have to be made out of materials that can stand that sort of high heat treatment. And there are some chemicals that will degrade under that. So we can't use autoclaving to sterilize them. We have to go back to filter sterilization or another strategy. Okay, so temperature is a really important way of getting rid of microbes, but it is not the only way. Now there is temperature treatment that's not quite boiling that is very useful in the food industry, and that's called pasteurization. Pasteurization generally is done between 60 and 80 degrees Celsius. So it's hot, but it's not boiling. And there are many more chemicals and products. Chemicals, I mean things that contribute to flavor, things that contribute to nutritional value that can withstand the temperatures of pasteurization that wouldn't be able to take boiling or autoclaving in an extreme example. Okay, so pasteurization is typically done at lower temperatures and for much shorter periods of time, right? So autoclaving is usually done for 15 or 20 minutes. Pasteurization is usually done for anywhere from 30 seconds to a couple minutes. So it's a much milder treatment, and the exact conditions can be worked out for whatever you're trying to pasteurize. Okay. What about the other extreme of temperature? Can you kill microbes by freezing them? And the answer is yes, to a certain extent. All cells can be damaged if ice crystals form inside them and usually killed. But we actually use cold as a way of preserving microorganisms when we add typically a cryopreservative or an antifreeze to them, like glycerol or DMSO, dimethyl sulfoxide. These things help to prevent the formation of ice crystals in cells. And I actually have strains of E. coli and Colibacter in my -80-degree freezer that are 30 years old, right? So they're very stable at those very low temperatures, as long as you've taken precautions to preserve them, okay? Now, another tool that is used often and maybe used more in the future is different kinds of radiation. So UV radiation, you're probably all familiar with because that's what you're trying to protect yourselves from in sunlight. It damages DNA. It does things like form thymine dimers, which are covalent bonds between bases that prevent replication. And so UV light is a legitimate strategy for microbial control. On the left-hand side here, you see a cell culture hood which has a UV light in top, and that is used to keep it clean and sterile in between uses or after uses, right? So that is important in the laboratory. There are practical ways of using UV light. This is a method called SODIS. The acronym is SODIS, SODIS, stands for solar disinfection. This is used in places that don't have effective water treatment. You can put your water in a plastic bottle, put it, expose it to sunlight for usually 2 to 4 hours. The sunlight provides UV light. Often the best way of doing it is to put it on top of a metal roof like this. And these sort of corrugated tin roofs are common in poorer countries, at least in some places. And so that roof also heats up. So you have a combination of heat and UV that is treating this water to disinfect any potential pathogens in it, whether they're viral or bacterial or whatever. It works as well as long as the water is pretty non-turbid. If you have a lot of dirt in there, then the UV light doesn't penetrate very well. UV light doesn't have great penetrating power. That's one of the problems with it with regard to disinfection. It works great on surfaces, but it can't go a long ways through the water. So another thing you can do is you can rotate these bottles around to try and get good coverage. But again, you leave it for a couple hours to get a good disinfection. Doesn't say here, and I honestly don't remember if it's on a later slide, but X-rays and gamma rays are also effective disinfection techniques, and those have much greater penetrating power, and so that can be used for sterilization or decontamination as well. There are lots of chemicals that can kill microbes fairly non-specifically or inhibit their growth. So there are a couple of terms to remember. So disinfection is using chemical methods to treat non-living surfaces to kill potentially infectious microbes, whereas antiseptics are chemicals that are used on living tissue, like preoperative, for example, or after a medical procedure to kill potentially infectious microbes. So the difference is just what you're using it on, non-skin, non-living material versus living material. Okay, here are some examples. You don't need to memorize all of these, but I would try and have a couple good examples for them. Ethanol and other alcohols are often used as disinfectants. They are sometimes used on skin as well. So this is kind of at the crossover between disinfection and antiseptics. Phenolic compounds, triclosan, oxidizing agents like sodium hypochlorite that you'd use in a swimming pool. Benzalkonium chloride is in a lot of the disinfectants. Like if you were in the lab right now, there would be a squirt bottle of disinfectant that had benzalkonium chloride in it and glutaraldehyde. So these are some different possibilities, and I'm sorry that these the structures got slid down a little, so these should be moved back up here because this is ethanol here, right? Okay, so there are a number of different types of chemical disinfectants. Should say that these usually work by, or they can work by oxidation of key components of proteins and lipids. They can work by acting as surfactants for disrupting membranes as well. So there are a variety of different ways they can, glutaraldehyde can work by cross-linking proteins. So there's different ways that these can cause damage to biological materials and kill cells, including microorganisms. Okay. So I believe this is the last slide. So something to think about is just some practical issues for destroying microbes or preventing their growth. How do you know what to do? Just, going through these quickly, thinking about what microbes are there. What are you trying to actually kill or prevent them growing? Are you worried about the presence of these very stable endospores, for example? What's the sort of microbial load that you're trying to deal with? Do you care if they all die? Do you need to actually accomplish sterilization, or do you just need to kill most of the potentially dangerous microbes? What is it that you're trying to treat? Is it a sturdy physical material? Is it a liquid? Is it a more fragile or labile? Are there key chemicals in there that can't be destroyed, or you lose the function of whatever it is you're trying to treat, etc. If it's a physical material, for example, can it stand up to heat? How sturdy is it going to be, etc. And how much do you need to worry about toxicity to humans or other life forms? For example, are there potential ecological consequences? Is there potential risk of toxicity to humans who happen to encounter it, right? So these are all things to think about. All right, so I'm going to stop there, and I will see you in class tomorrow.